Picture this: months of careful planning, late nights, and meticulous experiments, all leading up to a crucial moment. Then, in an instant, your membrane protein vanishes after adding the wrong detergent. All that effort, gone in a flash, simply because of a single overlooked decision. The result? Aggregated, inactive protein. Your experiment fails, and valuable time, resources, and even your motivation are lost. It's a setback that every researcher dreads, but it's more common than you might think. Membrane proteins are incredibly delicate, living in a fatty, protective environment within the cell. Their structure is finely tuned, and even small changes can have big consequences. To study them, we use detergents, special molecules that mimic the cell membrane and keep these proteins stable in solution, allowing us to extract and analyze them without causing damage. The right detergent preserves both the structure and function of your protein. If your detergent is too harsh, your protein unfolds and loses its activity. If it's too mild, you might not extract anything at all. Striking the right balance is essential. In this guide, we'll break down a clear, 5-step framework to help you pick the perfect detergent for your specific needs and experimental goals. You'll learn how to match your detergent to your goal, calculate the right concentrations, design the optimal buffer, and validate your results with confidence. Start by asking yourself, what's my main goal with this protein experiment? Are you aiming to preserve its function, analyze its structure, or simply extract as much as possible? Clarifying your objective is the first and most crucial step in choosing the right detergent. If you need to maintain protein function, like binding, transport, or catalysis, opt for gentle detergents such as DDM or LMNG. These help keep proteins active and stable, which is essential for functional assays. Avoid harsh detergents like SDS, which can denature proteins and destroy their activity, making them unsuitable for functional studies. For structural studies, you'll want detergents that form small, uniform micelles. DDM, C12E8, or OG are popular choices. These detergents help maintain the protein's shape for imaging or crystallography. Sometimes you'll need to screen several detergents or even swap them during crystallization trials to find the best fit for your protein stability and crystal growth. For proteomics, where extracting the maximum amount of protein is key and function isn't the priority, go for strong extractors like Triton X100. These break open cells efficiently. If you're doing chromatography, pick detergents that are compatible with your assay. Triton X100 or NP40 are usually safe bets and won't interfere with most purification steps. Ultimately, your experimental goal determines which detergent family you should use. Making the right choice here sets the foundation for your entire workflow and brings you halfway to success. Now that you know how to match detergents to your goals, let's move on to the next step in the process. Next, let's talk about how much detergent you actually need for your experiment. This is a crucial step that can make or break your results. Enter the critical micelle concentration, or CMC. This is the minimum concentration where detergent molecules start to form micelles in solution. Above the CMC, detergents self-assemble into micelles, tiny structures that are essential for solubilizing membrane proteins and keeping them stable in solution. To ensure complete solubilization, use 2 to 5 times the CMC, or a 10-to-1 detergent to protein mass ratio, whichever value is higher. This gives you a safety margin. Always check your detergent's CMC in the supplier's catalog or datasheet. For example, DDM has a low CMC, while OG's CMC is much higher. A low CMC means you'll need less detergent, which is not only efficient but also helps save on costs, an important consideration for any lab budget. Prepare a concentrated stock solution so you can add detergent without diluting your precious protein sample. This keeps your experiment consistent. Remember, CMC isn't fixed. It changes with temperature, pH, and salt concentration. Always match your buffer conditions to your experimental needs. Double-check your calculations and record every detail in your lab notebook or digital journal. Good records are key for reproducibility. Getting the detergent concentration right is absolutely crucial for experimental success and reliable results. Now, let's move on and design your buffer for optimal protein stability. Your buffer is your protein's new home. Get it right. Use a stable pH, 7.0, 8.5, with Tri-S-HCl or HEPs, 2050 nanometer. Add 150-300 nanometer NaCl to mimic physiological conditions. Avoid phosphate if working with metal ions. Always add protease inhibitors to protect your protein. Include a reducing agent like TSEP or DTT, 1,5 nm, for stability. Glycerol, 10-20%, helps keep proteins folded. Some proteins need native lipids or cholesterol analogs, add if needed. Test a few buffer variations to find what works best. A well-designed buffer can make all the difference. Time for solubilization. Be gentle. Add your buffer to the membrane pellet and resuspend carefully. No vortexing. Incubate at 4 degrees C for 30 minutes to 2 hours. Centrifuge at high speed to separate solubilized protein, supernatant, from debris, pellet. Collect the supernatant. This is your protein. Validate. Run SDS page and Western blot for yield and purity. Check for activity with a functional assay. Test stability by storing an aliquot at 4 degrees C overnight. For structural work, check mast cell size by size, exclusion chromatography. If all checks out, you've nailed your detergent choice. On to the next experiment. Thanks for watching. Don't forget to like and subscribe.